In the previous lecture, we examined how energy moves through marine food webs and how interactions among organisms shape ecosystem structure. We saw that energy captured through primary production moves through trophic levels and that the efficiency of this transfer influences how ecosystems are organized. Today, we focus on coral reef ecosystems, one of the most biologically diverse and ecologically complex environments in the ocean. Coral reefs provide a powerful example of how physical conditions, biological interactions, and energy flow combine to create highly structured and productive systems. What makes coral reefs especially interesting is that they exist in nutrient-poor tropical waters, yet support extraordinary biodiversity and productivity. Understanding how this is possible will be a central question for today's lecture. A coral reef is not simply a collection of organisms. It is a biologically constructed geological structure built over time through the activity of living organisms. The primary builders of this structure are reef-building corals, which secrete calcium carbonate to form rigid skeletons. Corals exist as colonies of individual polyps, each of which produces a cup-shaped skeleton beneath its body. As each polyp grows, it deposits new calcium carbonate, and as the colony expands, these individual skeletons fuse together into a larger structure. Importantly, when individual polyps die, their skeleton remains in place. New polyps then grow on top of these older skeletons, leading to vertical and horizontal reef growth over time. This process results in the accumulation of massive reef frameworks that can persist for thousands to millions of years. The image on the right shows a large coral that is estimated to be more than 1,000 years old. It is so large that it can be seen from space. Students that spend time at Goshen College's Marine Biology Station will see and learn that today's chain of islands that make up the Florida Keys is actually an old coral reef system that existed approximately 125,000 years ago, when sea levels were higher than they are today. The reef is therefore both a living system and a long-term geological structure shaped by biological activity. The physical structure that emerges is highly complex, consisting of ridges, crevices, and surfaces that create a wide range of habitats. This structural complexity is one of the primary reasons coral reefs can support such high biodiversity. It provides shelter, feeding areas, and attachment surfaces for a wide range of organisms. So when we refer to a coral reef, we are referring not just to living corals, but to an entire ecosystem built upon a biologically produced foundation that shapes the environment in which the organisms live. To understand coral reef ecosystems, we must begin with the coral polyp itself, which is the fundamental biological unit of the reef. Corals belong to the phylum Cnidaria, meaning they share key characteristics with jellyfish and sea anemones, including a relatively simple body plan and specialized feeding structures. Each coral polyp has a central mouth surrounded by tentacles. These tentacles are equipped with nematocytes, specialized cells that contain microscopic harpoon-like structures capable of injecting toxins into their prey. When small organisms such as zooplankton come into contact with these tentacles, the nematocysts discharge, immobilizing the prey. The polyp then transports the captured prey to its mouth, where it is ingested and digested. This feeding strategy allows corals to obtain nutrients and energy from the surrounding water, supplementing other sources of energy. At the same time, each polyp sits within a rigid calcium carbonate skeleton that it secretes. This skeleton not only provides protection and structural support for the polyp, but also contributes directly to the formation of the reef framework. It is important to recognize that corals are not passive organisms. They actively interact with their environment through feeding, growth, and reproduction. Their ability to both capture prey and build structure makes them unique among marine organisms and central to reef formation. One of the most important processes underlying coral reef ecosystems is the symbiotic relationship between corals and photosynthetic dinoflagellate algae, commonly referred to as zooxanthellae. These algae live within the tissue of the coral polyps, forming a highly integrated biological partnership. This relationship is a form of mutualism, where both organisms benefit. The algae perform photosynthesis, using sunlight to convert carbon dioxide and nutrients into organic compounds such as sugars. A significant portion of these compounds is directly transferred to the coral host, providing a major source of energy for the coral. This is critical because it allows corals to obtain energy not only from capturing prey, but also from internal photosynthetic production. In many cases, the majority of a coral's energy budget is supplied by these symbiotic algae. In return, the coral provides the algae with a protective environment within its tissues, as well as access to nutrients such as nitrogen and phosphorus derived from coral metabolism and waste products. These nutrients would otherwise be lost to the surrounding water, but are instead retained within the symbiotic system. This tight coupling creates an extremely efficient nutrient cycling loop. Nutrients released by the coral are immediately used by the algae, which in turn produce energy that supports coral growth. This efficiency is a key reason coral reefs can thrive in nutrient-poor environments. Coral reefs present one of the most important conceptual puzzles in marine biology, often referred to as the coral reef productivity paradox. The paradox arises because coral reefs are typically found in oligotrophic waters, meaning waters that are very low in dissolved nutrients such as nitrogen and phosphorus. Based on everything we've learned in the primary production lecture, we would expect such environments to support very low productivity. But yet coral reefs support some of the most diverse and productive ecosystems in the ocean. To understand this, we need to revisit the relationship between nutrients, primary production, and recycling. In most open ocean systems, productivity is limited because nutrients are quickly consumed by phytoplankton and then lost to deeper waters through sinking. This creates a system where productivity depends heavily on external nutrient inputs through mixing or upwelling. Coral reefs function differently. Instead of relying on external nutrient supply, they rely on internal retention and recycling of nutrients. The coral-algal symbiosis is central to this process. When the coral metabolizes organic matter, it releases nitrogen and phosphorus as waste products. In a typical system, these nutrients would diffuse away into the surrounding water. However, within coral tissues, these nutrients are immediately taken up by the symbiotic algae. The algae then use these nutrients for photosynthesis, producing organic compounds that are transferred back to the coral. So this creates a tight feedback loop where nutrients are conserved within the system rather than lost. This is fundamentally different from open ocean systems where nutrients are more easily lost from the surface layer. In addition, reef systems are highly efficient at capturing external organic inputs, including plankton and particulate organic matter. These materials are rapidly incorporated into the reef and recycled. So the paradox is resolved not by increased nutrient supply, but by increased efficiency of nutrient use. Coral reefs demonstrate that productivity is not only a function of nutrient availability, but also how efficiently those nutrients are retained and reused within an ecosystem. Coral reefs are structured into distinct zones that reflect gradients in physical conditions such as wave energy, depth, and light availability. These zones are not arbitrary. They are shaped by environmental forces that influence which organisms can survive in each region. The reef crest is typically the most wave-exposed area. Here, corals must withstand strong hydrodynamic forces. As a result, species found in this zone are often low-growing and robust, minimizing the risk of breakage. Seaward of the crest lies the fore reef, which slopes downward into deeper water. This region experiences reduced wave energy, but still receives sufficient sunlight for photosynthesis. As a result, it often supports high coral diversity and complex structures. On the landward side is the back reef, which is more sheltered and may experience greater sedimentation and variable environmental conditions. These factors can influence which species dominate in the region. Light availability is a critical factor across these zones. Because coral symbionts rely on photosynthesis, reef-building corals are generally restricted to well-lit environments. As depth increases and light decreases, coral abundance and diversity often decline. This zonation illustrates how physical conditions directly shape biological communities within coral reef ecosystems. Coral reefs are among the most biologically diverse ecosystems on Earth, supporting an extraordinary variety of species within a relatively small area. This high biodiversity is closely linked to the structural complexity of the reef. The three-dimensional framework created by coral skeletons provides numerous microhabitats. Crevices, overhangs, and open surfaces offer shelter from predators, feeding opportunities, and breeding sites. This diversity of habitats allows many species to coexist within the same general area. In addition to physical structure, biological interactions contribute to reef diversity. Competition for space is intense, particularly among corals and other sessile organisms. Predation also plays an important role in regulating populations and maintaining diversity. Symbiotic relationships are especially common on reefs. In addition to coral-algae symbiosis, many organisms engage in mutualistic interactions such as cleaner fish removing parasites from larger fish. These interactions create a dynamic system in which species are interconnected through feeding relationships, competition, and cooperation. The result is a highly complex ecosystem with intricate ecological relationships. Energy flow in coral reef ecosystems is distinct from most open ocean systems because of the combination of internal production, external inputs, and rapid recycling. To understand this, we need to integrate several concepts from earlier lectures. In the open ocean, primary production is dominated by phytoplankton, and energy moves through either the grazing food chain or the microbial loop. A significant portion of that energy is lost through respiration or exported to deeper waters. In coral reefs, a large fraction of primary production occurs within the coral itself through symbiotic algae. This means that energy production is localized and directly coupled to the organism that builds the ecosystem. Instead of relying on drifting phytoplankton populations, corals host their own primary producers. At the same time, corals are not exclusively dependent on this internal production. They also feed on plankton from the surrounding water, capturing zooplankton and a particulate organic matter using their tentacles. This introduces an external energy source into the system. So what makes reefs especially efficient is that very little energy is lost from the system. Organic matter is rapidly consumed, broken down, and recycled. This resembles an intensified version of the microbial loop we talked about before, but operating within a structurally complex ecosystem. The result is a system where energy is used multiple times before being lost, allowing reefs to support high biomass and diversity despite low nutrient availability. In essence, coral reefs maximize the retention time of energy within the ecosystem, which is a key principle underlying their productivity. You are likely familiar with the concept of coral bleaching, a phenomenon that results in the coral appearing to have been bleached white. But to really understand coral bleaching, we need to move beyond the visible symptom, loss of color, and focus on the cellular level mechanism that drives this process. Under normal conditions, the symbiotic algae within coral tissues perform photosynthesis efficiently. Light energy is absorbed by pigments and used to drive electron transport reactions that ultimately produce organic compounds. These reactions are tightly regulated and balanced within the cell. However, when water temperatures rise beyond the normal tolerance range of the coral algal system, this balance begins to break down. The photosynthetic machinery of the algae becomes unstable, particularly the components involved in electron transport. When this system is disrupted, electrons can accumulate and react with oxygen in unintended ways, producing reactive oxygen species, or ROSs. These are highly reactive molecules that can damage protein, membranes, and DNA. This creates a dangerous situation for both the algae and the coral host. The accumulation of ROS represents a form of cellular stress that can cause significant damage if not controlled. So in response to this stress, The coral expels the symbiotic algae from its tissues. This is a defensive mechanism intended to prevent further damage from ROS. However, it also removes the coral's primary source of energy. Figure A in this diagram shows coral at normal temperatures without any oxidative stress. But figure B shows significant production of ROS and eventual oxidative stress that requires the coral to expel the algae before there is too much damage to the coral. So coral bleaching is not simply a loss of color. It is a stress response driven by breakdown of photosynthetic processes under thermal stress. Once bleaching occurs, the consequences extend far beyond the individual coral. Because corals rely heavily on their symbiotic algae for energy, the loss of these algae has immediate physiological impacts. Without the energy supplied by photosynthesis, corals must rely more heavily on feeding. However, this is typically insufficient to meet their energy demands. As a result, corals experience reduced growth, reduced calcification, and decreased reproductive capacity. If stressful conditions persist and symbiosis is not reestablished, corals may die. When large numbers of corals die, the structural integrity of the reef begins to decline. The calcium carbonate framework remains for some time, but without living corals maintaining and building it, the structure will erode. This has cascading effects on the entire ecosystem. Many reef organisms depend on coral structures for habitat, for shelter, and even for food. As coral cover declines, biodiversity often declines as well. In this way, coral bleaching represents not just a physiological response, but a system-level disturbance that affects the entire reef ecosystem. It demonstrates how tightly linked biological processes, energy flow, and environmental conditions are within coral reef systems. I should mention that sea temperature rose above coral's threshold temperature for about 6 weeks during the summer of 2023 in the Florida Keys. The result is that some reefs such as Looe Key and Tennessee Reef experienced over 85% mortality of their coral. The result has been loss of sponges, loss of crustaceans, and of course loss of many fish on these reefs. Coral reefs represent one of the clearest examples of an integrated ocean system where biological, chemical, and physiological processes are tightly coupled. From a biological perspective, the system depends on symbiosis, predation, competition, and nutrient recycling. From a chemical perspective, processes such as calcification and nutrient cycling shape the environment. From a physical perspective, light availability, water motion, and temperature influence nearly every aspect of reef function. These processes are not independent. For example, water motion influences nutrient delivery and waste removal. Light availability affects photosynthesis within symbiotic algae. Temperature influences both metabolic rates and the stability of symbiotic relationships. Because these processes are interconnected, coral reefs are both highly efficient and highly sensitive. Small changes in environmental conditions can alter multiple processes simultaneously, leading to cascading effects through the ecosystem. At the same time, reefs are connected to broader ocean systems. They exchange organisms' nutrients and energy with surrounding waters, linking them to plankton dynamics, food webs, and larger scale ocean processes. Understanding coral reefs therefore require thinking in terms of systems, not isolated components. This perspective will be critical as you begin to observe and interpret reef environments directly in the field. Today we examined how coral reefs function as highly efficient and complex ecosystems. We saw how symbiotic relationships, nutrient recycling, and structural complexity allow reefs to sustain high productivity and biodiversity in nutrient-poor environments. These systems provide a powerful example of how biological interactions can shape ecosystem function and overcome environmental limitations. In the next lecture, we will shift our focus to open ocean habitats, where conditions are very different and productivity is often limited by nutrient availability. Comparing these systems will help us better understand how different parts of the ocean support life.